Hello everyone, wishing you all another beautiful, wonderful and an amazing day. In our previous class, we have seen an overview of biomicromolecules or biomolecules. So today we will discuss regarding the biomicromolecules. In our previous class, we have seen the flow chart of the biomolecules. What we are going to learn in this chapter, we had an overview. So today we will discuss regarding biomicromolecules. So before moving to that, we will have the analysis of chemical compositions. In the analysis of chemical composition, we will learn how we are going to get the micromolecules, how do we obtain macromolecules. So for that, first what we have to do, we have to take a living tissue. Living tissue, it could be some vegetables or it could be a piece of liver. And after taking that, we have to mix it with the trichloroacetic acid. And we have to grind it with the trichloroacetic acid, this piece of vegetables or piece of liver. After that, we will be obtaining a thick slurry. And this thick slurry, we have to stain through a cheesecloth. And here, we are going to get two fractions are being obtained here. So let us see what are the two fractions. What are the two fractions? That is acid soluble pool as well as acid insoluble pool. In acid soluble pool, we can see here the molecular mass will be of less than 1000 daltons. So that is said to be micromolecules. And in acid insoluble pool, that is more than 1000 daltons. And it is said to be macromolecules. That is said to be macromolecules. And here the examples of the acid soluble pools are amino acid, lipids, monosaccharides, that is sugars. And here in the macromolecules we have examples as protein, nucleic acid, polysaccharides and lipids. So you might have noticed in both lipids are there. How in both lipids are being obtained? So we can say that lipids are strictly micromolecules. Okay, lipids are strictly micromolecules. But when we analyze it, sometimes we may get a fraction in acid insoluble pool. That's what it is found in this acid insoluble pool also. So moving towards amino acids. What should be the essential elements in the amino acids? So we are moving towards amino acids. So what should be the essential elements in the amino acids? It could be carbon, it could be hydrogen, it could be oxygen, it could be nitrogen. So these four elements should be there in the proteins or amino acids. So these four elements, carbon, hydrogen, oxygen and nitrogen, these four elements are very, very important that should be there in the proteins or amino acids. And these are formed by the peptide bond. How the formation of the peptide bond is taking place? For that energy is being required. So these are linked by the peptide bond. How it is formed? Due to the energy is required for the formation of the peptide bond. And we know that around 20 types of amino acids are there. 20 types of amino acids are there. We will be learning the structure later on. Afterwards we will see. Now just let us see. See here the structure of amino acid. See, alpha carbon is present here. Alpha carbon, that is in between. Then one hydrogen is attached to it. One hydrogen is attached to it. Then one amino acid is attached to this group, and opposite to it carboxylic acid is attached to this group. One R, it could be of alkyl group, any of the alkyl group could be attached to here. And in H you can see G is attached, that is glycine. A in the CH3, that is alanine, and CH2OH ethanol, S is being attached, that is serine. Okay, so G for glycine, A for alanine and S for serine. So these are the three types of amino acids. This is very, very important when we learn about the amino acids. So amino acids, there are essential amino acids also, non-essential amino acids also. So let's learn about that. As I have told you, there are 20 types of amino acids. So essential amino acids are also there, non-essential amino acids are also there. So essential amino acids means it cannot be synthesized in the body of the animals. Essential amino acids are those amino acids it cannot be synthesized in the Then from where we will obtain it? We have to take it from the dietary protein. For the essential amino acids, we have to consume it from the dietary proteins. So here there are 10 essential amino acids. How we will learn? We can learn it in a tricky way. So here it is a TV-mill path. TV-mill path. In that way, if we break and learn, we can learn it. So T stands for tryptophan, V stands for valine, M stands for methionine, I stands for isoleucine, L stands for leucine, another L stands for lysine, then P stands for phenylalanine, A stands for arginine, and T stands for threonine, and H stands for histidine. Here we can see arginine and histidine I have marked with a special star. So these are semi-essential amino acids. Actually, it is not essential amino acid, it is semi-essential amino acids. Why I said that? Because when children grows and when the pregnant mother, they require it for the growth, this arginine as well as histidine. Rest all are essential amino acids. As I have told you, it can't be synthesized from the animals. We have to take it from the dietary protein. Now moving towards the non-essential amino acids. Non-essential amino acids means it can be synthesized in the body of the animals. It can be synthesized from the body of the animals except these 10. TV-mill path except these 10. Rest all are non-essential amino acids. We have serine, we have tyrosine, we have proline, we have glycine, we have alanine, we have asparagine, we have glutamine, we have cysteine, we have glutamic acid, we have aspartic acid. All these are non-essential amino acids. So I have told you essential amino acids we are not able to synthesize in the body of the animals, while non-essential we are able to. It can be synthesized in the body of the animals. What are the examples of serine, tyrosine, then proline, glycine, alanine, asparagine, glutamine, cysteine, glutamic acid, aspartic acid. All these are the examples of the non-essential amino acids. Then here we have another thing, that is Zwitter ionic form. So why do we call it as Zwitter ionic form of amino acids? Those amino acids that which is carrying the positive charge as well as the negative charge in the same amino acids, then we can call it as Zwitter ionic form of amino acid. Zwitter ionic form of amino acid or ionic acid we call it as. So if it is carrying one positive charge and one negative charge, you can see here NH3 plus amino group and carboxylic group. One positive charge and one negative charge. Another thing is that if we have to learn here is isoelectric point. What is isoelectric point? In which pH value it is being formed. In which pH value it is being formed, that is isoelectric पॉइंट. जिस pH वैल्यू में फॉर्मेशन हो रहा है उसी को हम क्या बोलते हैं? आइसोइलेक्ट्रिक पॉइंट बोलते हैं. सो आई होप यू माइट हैव गॉट द Zwitter ionic form as well as the isoelectric point. Next we will be learning few more amino acids that is being categorized here. We will go through it. As I have told you just now, amino acids are being categorized. Few more amino acids are being categorized here. What are they? They are acidic amino acids, basic amino acids, neutral amino acids, aromatic amino acids, and sulfur containing amino acids. So here acidic amino acids we have examples as glutamic acid and aspartic acid. Glutamic acid and aspartic acid are the examples of acidic amino acids. Then basic amino acids we have lysine as well as arginine. Basic amino acids we have examples as lysine and arginine. Once it was asked as a question, which of the following amino acids have positive charges at pH 7? So I do repeat, which of the following amino acids have the positive charges at pH 7? Which one has? That is basic amino acids. What are they? Lysine and arginine. So options will be given: aspartic acid, glutamic acid, lysine, arginine. So you have to write the basic amino acid, that is lysine as well as arginine. Then we have neutral amino acids. Examples are alanine, valine and glycine are the examples of neutral amino acids. Then we have aromatic amino acids. They are tyrosine, phenylalanine, tyrosine and phenylalanine. From tyrosine, what is happening here? Hormones are being synthesized. Okay, what is happening? Hormones are being formed by the amino acids. So melanin is being formed here, thyroxine is being formed here, adrenaline is being formed here. So melanin is being synthesized. Okay, the tyrosine will help to synthesize melanin, and thyroxine is also being synthesized, adrenaline is also being synthesized here. Then we can see here indole acetic acid that helps in the plant growth hormone. That is considered as plant growth hormone, indole acetic acid. Then amino acids that which contains sulfur. They are also very, very important. What are they? Cysteine and methionine. Cysteine and methionine are the sulfur containing amino acids. This is also very, very important question. Which are the sulfur containing amino acids? So moving towards lipids. Do you have any idea of lipids? Yeah, of course. We know that there are three types of lipids. What are they? They are monoglycerides, diglycerides and triglycerides. Okay, lipids are of three types. They are monoglycerides, diglycerides and triglycerides. So fats and its derivatives are combined and we call it as lipids. And the fats and its derivatives are combined, we call it as lipids. So we can say that lipids are generally insoluble in water. They are insoluble in water and they don't form polymer. No formation of polymer is taking place here, and large molecules containing an acidic group. Okay, large molecules will be containing an acidic group, and here R could be of alkyl group. It could be of any items can be attached to this R group, but it is soluble in organic solvents. So lipids are soluble in organic solvents such as acetone as well as chloroform. So they are soluble in organic solvents. They are acetone as well as chloroform. So now let us learn about the typical fat molecules, how it is being formed. So now let us learn how the typical fat molecule is being formed. What is typical fat molecule? They are triglycerides. Okay, how it is being formed? They are formed by 1 glycerol and 3 fatty acids. They are formed with 1 glycerol and 3 fatty acids. Sometimes it is often confused, 3 glycerol and 1 fatty. No, it is 1 glycerol and 3 fatty acids. So we can say that triglyceride is formed with a combination of 1 glycerol and 3 fatty acids. We can call glycerol otherwise as trihydroxypropane. And how it is being formed, this typical, what is glycerol and how this glycerol and fatty acids is being formed, where is it derived from? We know that it is simple lipids. It is derived from the simple lipids. So we have various types of lipids here, simple lipids, compound lipids, derived lipids. Now let us learn these lipids in detail. So simple lipids, they are divided into unsaturated fatty acid and saturated fatty acids. Compound lipids, they are phospholipids as well as glycolipids. Phospholipids where we can see lecithin, cephalin as well as cell membrane. Phospholipids we can find in cell membrane. Cephalin we can find it in the nerve tissue and all. Then derived lipids. Regarding the derived lipids, it is said that steroids and chromolipids are the derived lipids. Steroids, in the molecular biology it is said that most decorated micromolecules in the biology are cholesterol. When we see the structure, we can understand that it is so attractive. This cholesterol is the most decorated micromolecules in the biology. It is so attractive. When we see the structure, we can understand that why it is so attractive. It is said that most decorated micromolecules in the biology. Then chromolipid is terpene. Then oils are liquid at the room temperature when it is of low melting point, and fats higher when it is of higher melting point. Now let us see the differences between the saturated as well as unsaturated. Now let us learn the differences between the saturated fatty acids as well as unsaturated fatty acids. So unsaturated fatty acids, it contains one or more double bond or triple bond between the carbon atoms. So here it is formed with the double bond. Here it is formed due to the single bond. Here don't have any double bond or triple bond between the carbon atoms, while it is formed between the double bond. Here it is a single bond. Then we can say that it melts at lower temperature. Example is oil. Okay, liquid at the room temperature. Then here it it will be melting at a higher temperature, solid. It will be solid in the room temperature. While this is in the room temperature, it will be in the liquid form. And these are the essential fatty acids. Here it is the non-essential fatty acids. Where we can find the source of the unsaturated fatty acids? Plant fat. And here it is animal fat. In the saturated fatty acid, it is of animal fat. And we have examples in the unsaturated fatty acids as oleic acid, arachidonic acid, linoleic acid. And in the case of saturated, we have palmitic acid, stearic acid as an examples. So once again, let us see the differences between the saturated as well as the unsaturated. Unsaturated fatty acids, we can find in the carbon atoms, double bond and triple bond. Here don't have any double bond and triple bond. Here it will be melting at a lower temperature, while it is melting here at a higher temperature. And in the room temperature, it will be in the liquid form. Here in the room temperature, it will be in the solid form. And here we can find it essential fatty acids. These are the essential fatty acids. Here this is the non-essential fatty acids. And source as plant fat. Here the source will be of animal fat. And here we have examples as oleic acid, linoleic acid. Here we have the examples as palmitic acid and the stearic acid. So these are the examples. Now let us see the structures. So structure of fatty acid, palmitic acid, CH3, CH2, 14, COOH. Then here we have the structure of glycerol, here we have the structure of triglyceride, and here we have the structure of cholesterol, and here we have the structure of phospholipids. So each one we will see in details. Now, this is the structure of phospholipid. Okay, this is the structure of phospholipid. As I have told, this is the structure of triglycerides. So how the triglycerides are being formed, we have learned. 1 glycerol and 3 fatty acids. So as I have told you in the molecular biology, as I have told you, the cholesterol is the beautiful and attractive structure. So here you can see here, this is the structure of the cholesterol. So these are all very, very important. Tomorrow we will be learning about, in the next class we will be learning about biomicromolecules. Till that, bye. Wishing you a happy learning. Thank you. May God bless you all.